Hello everyone, welcome to the FEM meeting. Today we're glad to have Katrin Ackermann from St. Andrews to talk about pulse EPR at nanomolar concentrations. Katrin joined St. Andrews in 2012 and first worked on NMR protein. She then joined Bella's group and started using EPR for protein research. And now she is pushing the detection limit of pulse EPR down to nanomolar scale. So we're very looking forward to hear her talk. Katrin, the mic is yours.
Great. Thank you very much, Yuji, for the kind introduction, and also thanks a lot for inviting me to give this presentation today. So before actually going into the science, I just want to briefly talk about St. Andrews for those of you who haven't been here yet. So St. Andrews is a town in Scotland. We are situated at the east coast, about an hour's drive north of Edinburgh, and about 20 minutes south of Dundee. And it's quite a small town, really. That's sort of it. So we're about 18,000 population, and on top of that, we have about 10,000 students. And St. Andrews also has a lot of golf tourists, so it's globally known as the home of golf, and this is the famous Old Course and the Old Course Hotel on it. Apart from golf, the university is actually the main employer here in town. Quite an ancient university, but we also have some newer buildings. So this is part of the life sciences complex, and also where we have our biomolecular work done in the labs there. Right, so why nanomolar PDS? So PDS, pulse dipolar spectroscopy. So you can say if you just use some decent concentrations, you don't need to bother about nanomolar PDS, but it's not always a choice at what concentrations you can work. And also there's currently a drive in EPR really towards conditions that are more physiological. And that means not always, but quite often that we should be able to work in low concentrations. And also we know from our binding studies that you need to be somewhere in the range of the dissociation constant with your concentration. And again, that might mean that you need to be able to measure quite low concentration. So just a brief example, we are currently interested in a bacterial surface protein. I gave you here an electron micrograph from the citation here, where you see it as a sort of hair-like structures coming from the surface. And this protein is interacting with a protein from the host tissue. And if we want to do binding studies there at somewhat physiologically relevant conditions, we will need to go down quite low with the concentration. So clearly for us, sensitivity is key to accessing new science here. And one of the limiting features is the feasible measurement time. Now that might be quite different from lab to lab. For our case, it's about a weekend, so 48, 60-ish hours. So what we can just about measure in that time frame is what we say the lowest we can do. And basically, yeah. So just very briefly for the spin labels that we work with, we have the MTSL, that's our workhorse nitroxide label. And don't think I need to say much about that. Then we have the copper NTA that's been established by Sunil Saxena, and that is coordinated by 2 histidine residues. So that means it's a more rigid compared to the MTSL label. And so usually the distance distribution is a bit more narrow, or actually quite substantially narrower than with the MTSL. And also we have started using the SLIM label that was established by Olaf Schiemann and his group, and that has a particularly narrow spectral width, and that should be quite good for high sensitivity studies. For the model system, we use GB1. So that's really a great system to work with. It's small, it's rigid and robust, and it's been used by quite a lot of different groups in the magnetic resonance community. So for setting the stage, in our initial study, where we have been using the GB1 protein in our lab, we have been doing copper nitroxide rhythmy. And for the final measurements there that were used for dissociation constant determination, they were all done at 500 nanomolar. However, we actually have started much higher concentration at 25 to 75, depending on the construct. And then when we reduced concentration, we actually saw that the sensitivity doesn't suffer that much, really. And so that kept us thinking that we, probably most of us, actually underestimate the sensitivity that we do have. And that might be that because normally if we can, we use higher concentration. And only if we can't, that's then mostly actually that we use tricky systems. And then we need to go lower with the concentration. But that's not representative for what's the lower limit. And so we said if we want to rigorously benchmark the sensitivity for PDS measurements, we want to consider 4 things. We want to measure the standard experiments that we do routinely in our lab, and that is Peldor, Arcadia, and Whitney measurements. And we want to use our commercial, well-characterized Cuban spectrometer for these measurements. And we want to have a very well-behaved model system, such as the GB1. And we want to have a sample prep that is as consistent and reproducible as we can manage. So from that, we set out to look at different cases. And the first case we looked at was the copper-copper rhythmy. So we know now we can do the copper nitroxide rhythmy at 500 nanomolar. Can we do the copper as well? And you see here the background corrected traces and the corresponding distance distribution. And we get the expected distance peak out at 2.5 nanometers. Maybe you see that red line here, that's the modeling of the distribution, but we also get these additional probabilities. Some of them actually stay significant after the validation of the data, but they don't really, that's not corresponding to any true distances. And so to look at this a bit more in detail, we set up a very high concentration sample where we got really good signal to noise. And here you see that then these additional distance peaks vanish and they become completely insignificant upon validation of the data. And so that means that these distance peaks are in fact artifacts or distance noise because they tell poor data quality at the low concentration. And that means that we are here pretty much near the lower concentration limit for this case. This was also measured for more than 60 hours, so really as long as we can do. And that means that, in fact, comparing sensitivities, the copper-copper is less sensitive than the copper nitroxide experiments. And one reason for that is the comparatively lower modulation depth, because you cannot saturate both binding sites without drowning out your signal. And also the copper spectrum is quite a bit broader than the nitroxide, so you detect less of your spectrum, again, losing sensitivity. And so we went on to the next case, like the standard nitroxide Peldor at 500 nanomolar. And here we got quite good data out after an overnight measurement. So that's quite similar really to the copper nitroxide rhythmy. And you see here a bit of a shoulder in the distance distribution. That bimodal distribution is expected. It also shows a little bit in the model here. And we have seen that before for this construct that comes from an additional label confirmation. But still, we wanted to confirm that. And so again, set up a high concentration measurement where we got significantly increased distance resolution and of course, a much better signal to noise. And that clearly confirms the bimodality here for this measurement. And so here we can conclude that at 500 nanomolar for the nitroxide Peldor, we can still get out the true distance information, and that after just an overnight measurement. So we thought we should be able to get a bit lower with the concentration. And so we set up a 100 nanomolar sample, and the same dipolar evolution time was used for comparative reasons. However, we measured now for 2 days. You see the trace is a lot more noisy. However, we can still retrieve the mean distribution here. The mean distance, but the bimodality is completely lost now in the uncertainty. And that comes from the much lower signal to noise level, and that you then automatically use a larger regularization parameter. But this loss of bimodality has also been then confirmed using alternative processing approaches. So we conclude here that we are at the lower concentration limit for actually just the mean of the distribution, and we are already way beyond getting the true distance shape out, and that is quite a good agreement with recent data from Enrica's lab. So I don't want to spend too much time on this slide. I just want to say that we try to quantitatively assess and understand sensitivities that we got from these two different experiments, these two different cases, and that the numbers make sort of sense. And what we take from that, sort of the hypothesis we can say, is that the nitroxide Peldor should be quite close to the copper nitroxide RHYTHMI from the sensitivity. And so if we get this one down to 100 nanomolar, we should be able to get the copper nitroxide RHYTHMI to that concentration as well. And so that is what we tried next. So set up a pseudo titration at 100 nanomolar. This is the same construct as has been used for our initial study, where we did the same at 500 nanomolar. And here you see the different traces and corresponding distance distribution at that protein concentration with increasing copper. Each trace here is essentially a weekend, so each was about 60-hour measurement time. And the clear trend that I hope you can see is that we have an increasing modulation depth with increasing copper concentration. And also we get the expected distance distributions out for each of these traces. It's just that with the lower copper concentration, the uncertainty gets a bit higher. And this was also confirmed using DRNet for the processing instead of their analysis. So we conclude here that we can quantify modulation depth and reliably retrieve the distance distribution at that concentration for the copper nitroxide. And from these modulation depths, we can also use those for binding studies. So here you see the binding isotherm with the fitted dissociation constant, and that went all actually relatively smoothly. So we said we should actually be able to go down even a bit further. And so we went down by a factor of 2 in concentration to 50 nanomolar protein. Otherwise, complete the same setup. So again, 60 hours measurement time, because we can't really extend that much more. So expectedly, the traces are now quite a bit noisier. However, we can still get out the modulation depths, and they indeed follow the same trend as before. On the other hand, it's more tricky to get out the distance distributions, especially here when you go to the lower copper concentration. So either you have high uncertainty or spurious peaks that stay significant after validation. And that was also again echoed when we used DRNet for this. Some simply didn't run through, probably because of the poor signal to noise here. And so here we conclude that we can still quantify our modulation depths at 50 nanomolar concentration, but it's not really reliable anymore to also analyze the distance distributions. And that's also in line with the recent recommendations from the white paper that you shouldn't really trust distance distributions if your signal to noise is below 10. But since we can still get out our modulation depths, we can also still do our binding studies and get our dissociation constant, which is virtually the same as for the 100 nanomolar. So that's quite reassuring. But still, the error just gets larger. And so we think we are now at the lower concentration limit for the binding studies for this case. So now the question was, what can we do to increase sensitivity a bit more? So if you consider that when we do our copper nitroxide RHYTHMI, we actually only detect about 25% of the nitroxide line. So if we had a label with a very narrow spectral width, such as the tritol-based slim label, we might be able to detect the full line, and that should, in theory, give us a factor of up to 4 increase in sensitivity. And so we teamed up with Olaf and his PhD student Caspar to see what we can achieve there. And so we set out quite optimistically, just setting up a sample of 100 nanomolar, because we thought, okay, if we do that with a copper nitroxide, that runs over a weekend. So if we get somewhere in the range, the sensitivity increase that we think we should, then this would be a daytime measurement for the copper slim case. And indeed, you see here, after about 4 hours, we have relatively decent data. So that was quite promising. And we thought we should look at this label combination really a bit more systematically. And so we set up a whole series of samples ranging from 500 nanomolar protein down to 10 nanomolar protein. Everything was kept the same, all the measurement parameters. The only thing that we varied was the actual averaging time. And if you look here at the 500 nanomolar, that looks pretty crisp after 2 hours. So I found that quite good, really, especially if you consider that for the copper nitroxide rhythm, we measured that overnight, and I think the signal to noise wasn't as high as here. Also 150 looked fine, 50 was then overnight, and only the 10 and 25 nanomolar, after 2 days, the signal to noise, or the modulation to noise was under 10 still, but we could get the modulation depths out and you can get an idea how the distance distribution looked like. So overall, we think this is really a very powerful label combination. And when we then did some number crunching and looked at comparing sensitivities for the copper nitroxide versus the copper slim now, we got a factor of 3 improvement in overall sensitivity. And that was, remember that we said factor 4 would be ideal. So it's not too far off. Right, and with this, I would like to summarize. So we have the copper rhythm. First, we couldn't get lower than 500 nanomolar concentration for this case. That was mostly because of the lower affinity of the sheet side, which limits your achievable modulation depth. For the nitroxide Peldor, we went down to 100 nanomolar, but if you remember, we lost the bimodality there, so that affects distribution shape. For the copper nitroxide rhythm, we could go down to 50 nanomolar if we were only interested in the KD. So essentially retrieving modulation depths. If we also wanted to look at the distance distributions, we had to use the 100 nanomolar samples. So in fact, these two, like the nitroxide Peldor and the copper nitroxide rhythm, they are pretty similar in sensitivity. For the copper nitroxide rhythm, you just have, let's say, a bit of a smaller penalty to pay for optimizing the experiment at low concentrations. And for the copper slim rhythm, we got something like a 3-fold sensitivity improvement compared to the copper nitroxide. So it's really a promising combination. But of course, there are challenges that we haven't yet studied, like longer distances or more complicated distributions. So what's next? So for us, the next thing is to take samples to hyper here. That's the high sensitivity EPR spectrometer with the famous witch hats built by Graham Smith and his team in physics, and also use the AWG at Cuban there to see what we can do. And there's more developments out in terms of instrumentation, like the cryo-amp that has revolutionized NMRs now seem to come to EPR as well, and Mantas will get the IES Young Investigator Award at the RCESR in Leeds in a few weeks' time. And there's already the next generation slim label on the way from Olaf Schiemann and his group that is even more bioresistant and has improved relaxation properties. So that should all help with sensitivity further. But the good thing, kind of the take-home message, is that with the current concentration that we are now, especially for the copper slim combination, we are already in the same ballpark as many of the cellular proteins. And that means that the PDS binding studies at physiological concentrations should be within reach. And with this, I would like to thank Bela and past and present members of the board group, especially Josh for all his work here on the binding studies, and Hassan for keeping Cubant coming, and Olaf for collaborating with us on the copper slim, all the funding buddies. And thank you for your attention.
Many thanks for the nice talk, Katrin. I
Thank
see people clap. Is there any questions? No. I will start with the Hassan. Hassan, you have a question.
I do have very quick questions.
Yeah.
Do you expect to have any orientation selection in this system?
I think it depends very much on your label and the system used. So yeah, for some it should. I would be. You're referring to going to hyper with that, right? So I think it would be very interesting to look at the orthogonal labeling there and see what we can get.
Yeah, especially because you do have the copper, and
yes,
I think you should expect maybe to have some orientation selection. And in this case, depending on how they are or how the orientation is, so you should maybe expect more distance distribution depending. Yeah, I was just wondering if you have or you didn't look at all, but yeah, it was just a quick question.
Okay. I have a quick question as well. So when you do this low concentration experiment on hyper, what kind of difficulty do you expect?
So I have to say I haven't measured yet on hyper. So I expect that we will hopefully look at that together. I would expect that it's not that it should be that we should be able to take like the same samples really over and that we should be able to do direct sensitivity comparison with the hyper, and that potentially hyper wins, but let's see.
Okay, and another quick question, where you show the copper slim rhythmic results. I see that for 10 nanomolar and 25 nanomolar, so it doesn't really make a difference. It looks good for both of them, quite similar.
Yeah. Yeah. So if you just look at the numbers, the 25 is slightly lower than you would expect when you just look at the whole series. So I think it's just quite difficult at these concentrations with to directly say, oh, I expect this to be a factor 1.5 or 2.5 different.
Okay. Okay. Yeah, but in general, I think the result is very promising.
Yeah, I think so too.
Yeah. Okay. Is there any other questions?
Yeah, so it's quite difficult to adjust like the exact concentrations because we cannot, you start the preparation from a certain stock that you get as accurate as you can, and then you do your dilutions, but those you can't check anymore.
Yeah.
Yeah, so that's then difficult to adjust, but that's why it's important to have that sort of kind of robust comparative sample prep in between the different samples of a pseudo titration or in between the different constructs.
Okay, I see another 2 questions, Janet.
Hi, Katrin.
Hi,
Local questions. Very nice, thank you. I was wondering with this data here, how do the distance distributions compare for the NO and the SLIM? Sorry if you showed them and I missed it.
The distance distribution. What do you mean?
Well, the actual distance measured and the.
So this is for the SLIM.
Yeah.
And then we have these ones here for the.
Okay, so the MTS data is shorter distance and a narrower distribution.
Yeah, so basically for the SLIM, so I'm still waiting. I can't do the modeling yet properly, but that will come. So that is sort of in progress, that we can do the proper simulation what we would expect. But yeah, there is the copper is in the MMM and the SLIM is in the MTSSL without. So it's need to basically see that we can compare the expected distributions, but at the moment I can't do the full modeling for the copper slim case yet. But that's in progress.
Okay, thank you.
Hassan has another question.
Yeah, just a quick one, local questions. So you mentioned that you will use maybe in the future the AWG. So do you have in mind it's just the pale law, or extend that one to all the methods? Or so do you have in mind any experiments that you think that may help?
So I think we should. I think it should be possible to use the shape pulses that might help you with improving your sensitivity. But I haven't. I have used it for now, but not with the shape pulses yet. So I just want to basically use that as a start for. And I think we should try with Beldor specifically, but yeah, potentially also for the Redmi.
Okay.
And Fourier transform.
Another question from Yen. Go first.
Hi, nice talk. Thanks. I'm wondering, at those very low concentration, do you still see significant intermolecular interactions? And my question behind this is about the background. Could you imagine that you can even not use any background correction because you have so little intermolecular interaction? In other words, when you decrease concentration, do you see very flat backgrounds?
Not really. I mean, I think if we just use it as is, I haven't tried it, to be honest. I mean, you have that spin diffusion contribution from README, but I wouldn't. I haven't tried to just ignore background completely.
Yeah, I'm just wondering, because this is, I assume, something that brings a little bit of uncertainty, doesn't it? Like correcting an accurate background when you have such a high or such a low signal to noise ratio, then it becomes an issue, doesn't it?
And yeah, I mean, we have quite a robust protocol for validating the data. In fact, adding still noise to quite already noisy data. Okay. But that is all processed like that, like with 50% noise added on top.
Okay.
Yeah. So I think that's probably why it's to look quite interesting.
Yeah. Okay, thanks.
Okay, thanks for the questions, and let's thank again for Katrin's excellent talk. We'll see you in 2 weeks' time. Bye.
Thank you.